All right. Good afternoon, everyone. Welcome to the April edition of the Discover Lecture. My name is Alf Garcia-Bennett, and I want to welcome you to Macquarie University and acknowledge country by showing you this video.
Welcome. We acknowledge the traditional custodians of Macquarie University land, the Warramadigal clan of the Darug Nation, whose cultures and customs have nurtured this land since the Dreamtime. We pay our respects to elders past, present and future. We welcome people of all nations and all faiths. Kwai Bidja, Jamna Payala Janawi. Come here, we speak together. We have 60,000 years of archaeological evidence of Aboriginal habitation at Lake Mungo and 20,000 years in Ryde. We have great antiquity. Today, hundreds of Aboriginal and Torres Strait Islander people graduate from Macquarie University. The Darug Nation had famous leaders such as Chief Yarramundi, Naraginji, Colby and Maria Locke. Many of the descendants of these Darug people live today amongst you. We celebrate with you our ongoing attachment and custodianship of this country. We celebrate the achievements of Macquarie University.
Okay, today's speaker is a senior research fellow at the Australian Research Council Training Centre for Facilitated Advancement of Australia's Bioactives, which we shorten to FAAB. I have the honour of being the current director of FAAB. So FAAB aims to change the way we think about bioactives. We are collaborating with large companies, with startups, in order to really understand the bioactives in the products that they develop. So we discover bioactives with them. For example, we are studying how fish oils can reduce inflammation in the brain. We innovate in analytical methods with them. For example, we are trying to uncover the science of the bioactives in broccoli leaf. So bioactives are all of those molecules that have a nutritional, healthy value when you take them. And because most of the supplements and bioactives that we take them as a food source or orally, it is very important to understand how they interact with the gastrointestinal tract. That is the topic of Hasinika's research. And her research team and herself has innovated in bioinformatics and sequencing in order to understand how gut microbiome can be used to prevent diseases like colon cancer or neurodegenerative diseases. Hasinika's research and that of her team is considered to be excellent, first class. And evidence of this is the award of the John Christian Young Microbiologist Award. Hasinika will talk today about her current research in gut microbiome and how it relates to some of the work she does at FAAB, but also external to FAAB. So over to you.
Can you hear me? Amazing. Thank you so much, all, for that introduction. Thank you all so much for coming along today. I really appreciate it. Thank you to our Faculty of Science Outreach team for the invitation to talk, and to FAAB for the opportunity to talk. So I look forward to talk to you about the gut microbiome, how it's developed, its link with diseases, and how we can change the microbiome in a way to either prevent or treat these different diseases. I'll touch on the work that we do at FAAB and also the work that we do outside FAAB as well, broadly at Macquarie University. So I'll add a little bit more to what Alf mentioned about FAAB. FAAB is an Australian Research Council funded research centre, and we sort of provide a platform for academic researchers to collaborate with industry partners. We work with around 15 different leading Australian companies. We have around 19 research students working with us, and 3 of the amazing students who are working with me on gut microbiome projects are going to talk to you about the work that we do at FAAB. So like Alf mentioned, we are focused on identifying, enhancing the delivery of bioactive molecules. So at FAAB, we assess the impact of these bioactive molecules as one of the measurements of that being gut microbiome, and I lead the research on that. So before we start, I'm sure a lot of you might have heard that we are more microbial than we are human. So in a grown adult, in an average, we have around 30 trillion human cells. In comparison to that, the microbial friends that inhabit our body, there's around 40 trillion microbial cells. So we are actually only 43% human if you think about it. We become even more insignificant when we think about the genetic material. As humans, we have around 20,000 functional genes, and our microbiome, the microbes that live in and on us, they have around 2 to 20 million genes. So this sheer number, their sheer genetic potential, makes them, like what they do, really impact our health and well-being, their functions, the metabolites that they produce. So where do these microbes live? They pretty much live everywhere on our body. There's microbes living on our hair, skin, stomach, colon. As shown in this diagram here, the exact composition of the gut microbiome or microbiome that's living in these different body sites do vary depending on the nutrients these body sites provide or the availability of oxygen. So for example, microbes that live on our skin is quite different to those living in our colon. Mainly because, as you can imagine, the skin is exposed and those microbes can tolerate oxygen, whereas in our colon, it's mostly oxygen-free. So bacteria that's living in the colon, they don't like oxygen, so they thrive when there's no oxygen. So given those selection criteria, the exact composition of the microbiomes in these different body locations do vary. The vast majority of the microbes that are living with us, around 95% live in our large intestine. And we refer to them, and also not just the microorganisms, but their genetic material as the microbiome. So that's the definition of microbiome. So we usually talk about bacteria when it comes to microbiome, which is fine because they form the majority of the microbiome, but there are smaller proportions of other microorganisms as well, like yeast, fungi, viruses, archaea. So gut microbiome contains a lot of bacteria. To give you a perspective, in a grown adult, in around 1 millimeter length of the colon, there's around 20 to 30 billion microbial cells. And our colon is around 1.5 to 2 meters long. So you can imagine the sheer number of microorganisms that's living in us. So there's been a recent increase in our awareness in gut microbiome research. If you go to the supermarket, the health food aisle is packed with probiotic products, prebiotic products, and you hear about gut microbiome on social media, mainstream media, it's pretty much everywhere. This surge really happened around, I would say 10 to 15 years ago. And it really comes down to that we really didn't have the technology 20 years ago to study microbiome. Especially gut microbiome, they're difficult to grow in the lab because, firstly, because they don't like oxygen, but we now have techniques to overcome that. But also because they're different microorganisms, they have different nutrient requirements, so it's difficult to study them as a whole if you are going to culture them or grow them in the lab. So these techniques called omic techniques, they actually accelerated gut microbiome so much in the last 10, 15 years. So we don't necessarily culture the gut microbiome, we study the community as a whole. So we extract the DNA from the community and we read the building blocks of DNA. It's called DNA sequencing. So this approach is called metagenomics. And similarly, we can take all the RNA out of the community and read the makeup of that RNA. That's called metatranscriptomics. We can analyze all the proteins in the community, metaproteomics. Metabolomics is where we would analyze all the compounds, all the molecules this microbial community produces. So these sort of shotgun approaches where we're not really isolating a single strain of bacteria, but we're just looking at a community as a whole, has really advanced microbiome research. So I'm just going to dive in a little bit deeper to DNA sequencing, especially because it is, I would say, the mostly used technique. If you look at a gut microbiome paper, I would say 95% of the time they use DNA sequencing. So this comes down to this technique called next generation sequencing. What it essentially is, so in us, in a plant, in a bacteria, our DNA is made with 4 bases. So adenine, thymine, guanine, cytosine. And they're present in different sequences throughout our genome. So this technique can read these building blocks. So you can imagine when there's trillions of microorganisms living in our gut, that produce a large amount of data. And sometimes this is terabytes of data. So we need the computational power to analyze them. So we do have that now to sort of facilitate microbiome research. I'm going to dive in a little bit deeper. So with DNA sequencing, what happens is we take the DNA out of the microbial community, and there's 2 ways of looking at it. The first way is called amplicon sequencing, where we're just looking at a single gene. They're called marker genes, and they're present in all bacteria, and we just sequence that piece of gene. With this technique, we will get information about who is present in the sample and at what proportion. The second technique is metagenomics, where, like I mentioned, we just take all the DNA out and we sequence everything. So all the DNA from bacteria, everything from fungi, viruses, everything that you get from the gut microbiome. This obviously generates a lot of data and it's much more costly, but it's advanced in that it not only tells you about the composition, the proportion of different microorganisms, but also their functions as well. So with this development in techniques, we now know microbiome is really important for our health and well-being, starting from helping us to digest food that we can't otherwise digest. So we as human beings, we don't have all the enzymes needed to digest dietary fiber. So they go to the colon undigested, and our gut microbiome digests it for us. This process produces metabolites, especially metabolites called short-chain fatty acids. We know, have significant impacts on our body, on key organs in our body. These molecules sort of regulate how fat and glucose is being metabolized, and also they help with the communication between our gut and brain. Gut microbiome also helps to synthesize vitamins that we can't otherwise get, vitamin K, vitamin B group. Another key function of the gut microbiome is that its impact on our immune system. It actually helps the development of our immune system. It will remove pathogenic bacteria from the gut. So each one of us, we have around, it's a big range, 300 to 1,000 different species of microbes. But what is interesting is that each one of us, we have a unique gut microbiome. If you're from the same family unit, maybe I think there's similarities, but you still have your unique microbiome. It's quite different from individual to individual. This comes down to a few things, starting from birth. So in utero, there's impacts on the microbiome from the mother, also the health of the mother on the development of the gut microbiome. Now, the biggest exposure to microbes happen during birth. So depending on the birth mode, the exact microbes babies get exposed to do vary. So if it is a vaginal birth, they get exposed to lactobacillus. And if it is a cesarean section, the first microbes they get exposed to are skin microbes. The next big thing is milk consumption. So lactobacillus, bifidobacteria, these bacteria can digest milk, especially milk oligosaccharides. So it is a neat system in that regard. The other big introduction is through when we introduce solids to babies. That's when they start to develop a microbiome that's capable of digesting dietary fiber. So around 3 years of age, babies develop a more adult-like microbiome that is more stable, diverse, and they say the first 1,000 days of life is very, very important for the development of the microbiome for that. So impacts of antibiotics, host genetics, environment, parents, whether you have siblings or not, whether you have pets or not, all these impact the development of the microbiome. As adults, our microbiome is quite stable and it's diverse. However, things like drastic change in the diet, aging, diseases, use of antibiotics, these can impact the microbiome. They can change the microbiome. So our microbiome is quite dynamic. It keeps changing, the compositions keep changing. So the gut microbiome is in constant communication with us. It happens through metabolites like short-chain fatty acids. It can even happen through neural pathways when it comes to the gut-brain axis. The vagus nerve that runs from the colon to the brain actually does that communication. Immune system is another way the gut microbiome communicates with us. Hormones and gut barrier being another mode of communication of the gut microbiome. Gut barrier, in fact, is one of the key research focuses here at Macquarie University. So the interior of our gut is made with this sticky gel-like mucus layer. And this mucus layer is made with proteins that are highly sugar-coated. Gut microbes love these sugars. So Lauren will talk to you more about these sugars towards the end of the presentation. Gut microbes use them as food, and they also use them as sites to adhere or stick to. So who sort of adheres to the colon actually shapes the composition of the microbiome. So when the microbiome, mucus layer, everything is in balance, everything works beautifully. And the mucus layer acts as a barrier between us and our gut microbiome. Now, with different diseases and even reduction in the intake of dietary fiber, this is severe reduction, gut microbiome turns into our mucus layer as food, and they start eating those sugars. And that the mucus layer then becomes thinner, and when the mucus layer, the barrier between us and the gut microbiome becomes thinner, microbiome and us come in contact, and that's the beginning of a lot of inflammation and leaky gut type conditions. So like I mentioned, when the gut microbiome is working beautifully, imagine a rainforest. It's an ecosystem, it's working well. But the external things that we introduce, especially as a part of the modern lifestyle, like highly processed food that is low in dietary fiber, high in fat and sugar, excessive use of antibiotics, physical inactivity, stress, all these can impact the microbiome towards making it less diverse, changing the composition. So I just want to draw your attention to this study that's published a few years ago now. What they did was they looked at the microbiome across a lifestyle gradient. So they looked at these tribal groups that live in Nepal, and some of them were hunter-gatherers, some of them were settled, some of them were farmers, and they compared their microbiomes with the USA modern microbiome. This plot here, one with the dots, individual dots show the microbiome of a person. So the gist of it is closer two dots are, their microbiomes are similar. Far apart two dots are, their microbiomes are different. So the striking thing here is those orange dots. They are the USA microbiome. They're quite different to the rest of them. But if you look at it, there's sort of a shift towards that. And that sort of follows the change in their lifestyle. So as the lifestyle sort of gets more modern, the microbiome tends to shift. And this becomes more apparent when we look at this diagram. I'll break it down. So this is a heat map. The rows show you the bacteria, and the columns show you individuals. So the color, darker it gets, it shows those bacteria have a lower abundance. I want to bring your attention to this group of bacteria. They're actually not present in the USA microbiome. So as the lifestyle gets more and more modern, these microbes tend to disappear from the microbiome. And these are fiber-digesting bacteria in the gut. So it's quite alarming with these modern lifestyle changes on how the microbiome tends to really change and become less diverse as well. So the modern lifestyle and the microbiome changes that happen with it has been associated with a lot of diseases, starting from neurodegeneration, diabetes, obesity, colorectal cancer, and there's a long list. I would like to point out two of the diseases that at Macquarie Uni we look at closely. The first one being colorectal cancer. So colorectal cancer is the second most common cancer in Australia, and it causes more than 5,000 deaths annually, just in Australia. Development of colorectal cancer, as shown here, is there's stages to it. The first few stages, it's growth of abnormal cells in the colon, and over the years, they become cancerous. However, the first few steps, they're not cancerous just yet. So we know that there's genetic changes, there's structural changes that happen in the colon with colorectal cancer development. We now know that there's microbiome changes happening with colorectal cancer development. And there are microbiome changes that are specific to each stage of colorectal cancer as well. Now, also, the microbes that sort of adhere or stick to the tumor is different to those living right adjacent healthy tissue. So with this knowledge, what can we do? So the fact that there's microbiome changes very early on, maybe that could be a marker for early diagnosis, and maybe microbiome could offer ways to prevent colorectal cancer development. It could be a marker for predicting cancer reoccurrence. Also, there's reports showing now that microbiome changes can also enhance the efficacy of cancer treatments as well. I have one example to show you here, which is showing the use of microbiome in combination with more conventional ways of testing or diagnosing colorectal cancer, the fecal test, and adding microbiome as an extra analysis, how that improves the detection rates of the current tests. So the black bars in this diagram here, they are when the traditional test is being complemented with microbiome analysis. The increase in the detection rate, particularly in these early stages of cancer, it's quite important because the earlier we can detect colorectal cancer or any cancer for that matter, there's higher survivability. So if we can enhance the detection rate early on using microbiome as a complementary tool, it is a great way of reducing the cancer burden and improving the quality of life for many. The other disease and microbiome link that I want to touch on is neurodegeneration and gut microbiome. We at Macquarie University, we study a disease called Machado-Joseph disease. It is a genetic neurodegenerative condition. It causes movement disorders, and people with this disease, within few years of diagnosis, they become wheelchair dependent. It's quite similar to Huntington's disease and even Parkinson's disease in terms of symptoms. It is important to Australia, especially because the highest prevalence is seen within Aboriginal communities in Australia. So we did look at this disease and the gut microbiome in an animal, a mouse model of the disease. What you can see here is a sequence of events in male and female animals. What we saw was that the gut microbiome of these animals changed well before any of the known movement symptoms appeared or were detected in these animals. We also looked at gut transit time, so it's the time for food to traverse from the mouth to the other end, and we saw that there's faster gut transit time, especially in male animals, even before the symptoms were detected. Interesting is that we didn't see this observation for female animals. This is not one observation in isolation. This has been seen in neurodegenerative conditions quite often. Also with Parkinson's disease, Alzheimer's disease, there's gut microbiome changes that happen well before any of the symptoms are detected. And in Parkinson's disease particularly, there's gut transit time changes happening years before, in people, years before the symptoms appear. So our next step in this particular disease model is to take it to human studies to see whether we see a similar change in humans as well. So, like I said, there's early microbiome changes in mice with this particular disease, Machado-Joseph disease, and the healthy controls. One significant finding is that animals with the disease do have a lower abundance of microbes that can produce this short-chain fatty acid called butyrate. So it's interesting, and we are looking into this more to supplement the gut microbiome of these mice with butyrate to see whether that can help with this disease. So I think I just touched on this briefly in the previous slide, but the question is, can we restore the gut microbiome? Can we take it from this less diverse, changed composition? Can we improve it and see a health outcome in humans and animals? The first answer is, have a healthy diet. Have more vegetables, fruits, nuts, legumes. It's a great solution. But it is sometimes difficult to implement. And maybe the microbes are missing in the gut. We really have to reintroduce them into the gut. The other thing is the time of the day that we eat. So this is an area that we have studied at Macquarie University. we do know our body is in sync with the light and dark cycle. So is our gut microbiome. There's changes in the composition of the microbiome, their functions, depending on the light and dark cycle. So eating when we are meant to eat, the light cycle, is really important for the health of the gut microbiome. So when, healthy lifestyle is not enough to maintain gut health, there are other options that's available. The first one being probiotics. So this is essentially we're seeding the microbiome with live beneficial bacteria. Probiotics can be found in your yogurt, fermented food, and a lot of the probiotics are available in the supermarket. If you go to the health food aisle, even health food store, you might see a lot of products, probiotic products. The way these things work, firstly, they compete with the gut microbiome or pathogens or harmful bacteria or microorganisms for food and space in the gut. And then they eliminate the harmful ones from the gut. Now, for this to happen, it's important that we consume a large number of these bacteria, probiotic bacteria, and that they're alive. And Joanna will talk to you more about it in a bit. These probiotic bacteria, especially Lactobacillus bifidobacterium, they are known to produce short-chain fatty acids. They're known to strengthen the gut barrier as well. So the second technique that I want to talk to you about is prebiotics. So this is essentially feeding our gut microbiome. So they love dietary fiber, like I mentioned earlier. So dietary fiber being one of the key ways that we nourish our gut microbiome. So there's dietary fiber supplements, again, available in abundance. The way that they work is through escaping our digestion, going to the microbiome, and they digest it for us and produce things like short-chain fatty acids. These short-chain fatty acids, like I touched previously, they have been shown to impact a lot of key organs and tissues in our body. Another thing that I want to point out is there's now postbiotics, which are essentially the bacterial metabolites being used as supplements. So like we're doing it with neurodegeneration, using, say, for example, short-chain fatty acids directly as a supplement. So those are called postbiotics. There's also combinations of prebiotics and so dietary fiber and probiotics. Those are called symbiotics. So there's multiple options available already in the market, and they're being studied in academic settings. The third method is fecal microbiome transplants. So as the name suggests, it's about taking the fecal microbiome from a healthy donor and transplanting it in a person who has an infection, particularly infection with Clostridium difficile. It's currently being used for that, and it has a really high success rate, around 90%. There's obviously a lot of screening that is required for this, but it is working for Clostridium difficile infection, and it's been studied as a way of changing the microbiome when it comes to inflammatory bowel disease, inflammatory bowel syndrome in research settings. Okay, with that, I'll pass on to Joanna to tell you more about the research that we do at FAAB in terms of making probiotics better in traversing the digestive system.
Hi all, I'm Joanna, and I'm excited to talk about my PhD project, which is in collaboration with Factors Group Australia, a company that deals with supplying probiotic products. So let me first begin with what are probiotics. As Hasinika has previously mentioned, probiotics are essentially live microorganisms, such as bacteria, that confer health benefits to the host. Probiotics are available to the consumers either in the form of supplements such as capsules or in the form of foods such as yogurt and drinks such as Yakult. Now, for probiotic bacteria to confer their health benefits, they must survive the harsh conditions of the digestive system so that they can successfully reach and multiply in the gut. However, during their journey through the digestive tract, they are exposed to conditions that can potentially kill them. Once a probiotic is consumed, the first digestive tract stressor it comes across is the acidic conditions of the stomach. So as shown in the plot over here, I have exposed an actively growing probiotic bacteria that is lactobacillus to pH 3.0, which is the acidic conditions in the stomach. And this has shown that their viability was reduced by more than 70%. After their travel to the stomach, they then move into the small intestine where they are exposed to bile salts. Now, bile salts essentially is involved in breaking down dietary fats, and they can also degrade probiotic bacteria as they contain lipid bilayers in their cell membrane. So if we just look at the plot again, I have exposed lactobacillus to biologically relevant 0.3% bile salts, and once again, that drastically reduced their ability to survive by more than 80%. So the current strategies to improve their ability to survive their journey through the digestive system is by creating capsules to provide a protective covering, such as giving it a buffering effect. And secondly is by selecting bacteria or bacterial strains that can survive the harsh condition of the stomach and the small intestine. However, these strategies alone are not enough to improve probiotic efficacy. So I am looking at components such as metabolites from the yeast, which is also known as postbiotics, and prebiotics, which are essentially carbohydrates or sugar molecules that can be added to the capsules to improve their ability to survive during the digestive system. So my work at FAAB is to look at whether prebiotics can enhance probiotic bacteria's ability to survive the digestive system. And based on that is to come up with unique prebiotic, postbiotic, probiotic combination. Now, in the gut, yeast and bacteria naturally interact with each other, and they feed on each other's byproducts, such as metabolites, for their survival. Therefore, I am using yeast metabolites as a potential strategy. Based on my data, we already know that addition of yeast metabolites to lactobacillus has helped them withstand the acidic condition in the stomach better. So I'll be further studying the role of yeast bacteria interactions in supporting probiotic bacteria's ability to survive the digestive system. Ultimately, this knowledge will be translated in producing probiotic products that can help us achieve maximal health benefits. So that's all from me today. Anna will now talk about prebiotics and its role in pet gut health. Thank you.
Thank you, Joanna. Hello, everyone. My name's Anna, and I'm a PhD student at FAAB, and I'll tell you a little bit about what I do. So, hold on. There we go. Okay, so In Australia, there are about 29 million pets, and 50% of those are dogs, 30% are cats. And with such a large pet population, it's no surprise that they've become cherished members of society, often referred to as family members. And so because of our deep connection with our pets, there is a lot of time and effort invested in improving their quality of life, from grooming to enrichment, things like doggy daycare. But one of the major ways that we focus on improving their quality of life is through their diet. And pet diets have evolved beyond simply meeting basic nutritional requirements. They're now formulated to support specific health outcomes by incorporating different ingredients that have known health benefits. And dietary fibre is one of those ways. So what do we actually know about dietary fibre in pets? Well, a lot of what we know about dietary fibre comes from an extensive research field in humans that tells us that it's really good for us. But, so while we have some similarities in our digestion, pets are not small humans, contrary to what some people believe, and species differences do matter. And so what you can see is a huge contrast of an extensive field of research in humans compared to the few hundred of studies in pets. And so we add dietary fibre to our pet food. And if it's going to be an important role in their diet, we should first look at if what we see in humans is actually applicable to pets. So the purpose of a diet is to provide energy, nutrients, support growth and well-being, as well as prevent disease. And to access these benefits, we need to unlock the nutrients. And so if you think about your food as having different padlocks, and each of these padlocks guards a specific nutrient group, such as protein, carbohydrates, fats, lipids, vitamins, all of it. So humans and animals come equipped with a set of keys to unlock these padlocks, and they can do everything except dietary fibre. So dietary fibre, as Hasinika mentioned, travels through your gut until it reaches the colon, where it comes into contact with the bacteria that have the missing key. And so now we can unlock the health benefits of dietary fibre, which is, as Hasinika mentioned, the short-chain fatty acid metabolites. And so at FAAB, I look at addressing this in two different ways. So in the lab, I grow bacteria from the gut in the presence of different types of dietary fibre. And I do so in a big chamber that has no oxygen. And I look at what the microbes are doing with that dietary fibre by looking at their DNA and then also looking at the different compounds that they produce. And then the second way is in real life. I'd run a diet study where dogs consume a specific type of fibre, and I do the similar thing and look at their gut microbiome, but I also measure their health outcomes to see how that impacts a living system. Because obviously a bottle versus a real life dog are very different systems. So these two methods together help us to first identify, well, what does the dog gut microbiome do with dietary fibre, but it also allows us to look at the health outcomes as well. So yeah, that's my project. Now I'm going to pass you over to Lauren, who's going to tell you about her work at FAAB and fecal microbiome transplants.
Hi, so I'm Lauren, and my work at FAAB, as Anna kindly mentioned, is on fecal microbiome transplants and the sort of relationship that it has with the mucus in our gut. So we've already touched on a lot about how to keep our gut healthy. We've spoken about prebiotics and probiotics. But what happens when things go wrong in our gut? What actually happens when our good microbes go missing? And we've touched about why this can happen. This can happen with things like diet, antibiotics, infections, even stress. And when it does happen, we can start to struggle to digest our food properly. Our gut can become inflamed, it can become irritated, and we can become more tired, we can have a lot of problems downstream. So the big question is, how do we get these good gut microbes back? And an answer to this, as Hasinika's already touched on, is by fecal microbiome transplants. And so we know that these can reshape and rebuild our microbial communities. But there's a sort of a missing part of this story, because as Hasinika's nicely touched on, yes, we know that we can put the microbes back into our gut, but how do these stay there? We know that we can transplant them in, and we know that some will stay, and we know that some won't. And why is that? And so that's a big part of my research. In our mucus, we have these special sugars. We call them O-glycans, but don't worry about that too much. Our microbes can actually attach to these O-glycans. And we think that it can influence which microbes thrive and which ones don't. So think of mucus sort of less as slime that covers our gut and more as a home for all these little microbes. And so here's where the mystery starts. We aren't very well. We have a fecal microbiome transplant. And we know that some of these microbes will stay and some of them will go. And so we think why this is because we put nice ones in that our body uses, and they can attach to these docking stations, these O-glycans, and they can stay, they can find food, and they can thrive. And others don't have these receptors to bind to. And so within the coming days or the coming weeks, they're flushed out of our system. And that's where my work starts. So we look at two different models. We look at the pigs, and then we take it back into the lab. In the pigs, or in the piglets, as piglets are weaned from mother's milk onto solid food, there's great distress to both the piglet and to the microbiome, and it causes diarrhea. And so we give the piglet fecal microbiome transplants, and we have a look and see in the untreated as well as the treated which microbes and which sugars stay, which ones change, and how the two groups are different from one another. And in the lab, we can take sugars and we can take key bacteria and we can make them interact with one another. We can take sugars away and we can decide which sugars are essential for colonization. And so that's sort of the gist of my work, sort of when the sugars change, who stays and who goes. And I'm going to pass back over to Hasinika, and she's going to talk a little bit more about the future of microbiome therapeutics.
In the next couple slides, I'm just going to wrap up. Firstly, talk about where gut microbiome therapeutics are headed to. So, like we mentioned, we have prebiotics, probiotics, fecal microbiome transplants as ways of changing the gut microbiome. But a common feature of all these, like these three techniques, is that their impact is untargeted. So if the aim is to improve the overall health of the gut microbiome, they're great ways of doing that. But there are more targeted techniques that are currently being researched, so they're not really available for general consumption yet, but they're being researched, and I'll tell you a bit about that. The first one is this concept called microbial consortia, and this particular one is actually being marketed, and there's products around this concept. So when it comes to a probiotic product, a lot of the products that you'll see in the market these days, they do have a number of different bacteria. They usually belong to lactobacillus or bifidobacteria. However, there's around 10 or even more different bacterial strains in a single capsule. The way that they're typically made is that they're grown individually and they form into a powder to last longer. And then these bacteria, these powders are mixed together. Now, with the concept of microbial consortia, they're grown together, they can interact with each other. So there's products that are available for that, and this was sort of introduced initially as an alternative to fecal microbiome transplants. But this is a concept of, it sort of sits between untargeted and targeted microbiome therapeutics, but it's an option that's been further researched. The last three are, some are available when it comes to the single strain strategies, especially in research settings, but phage therapy and synthetic biology applications are not really available for our consumption yet. Single strain live biotherapeutics, or NLBT, non-live biotherapeutics, these are using a specific strain of bacteria from the gut to deliver a known health benefit. So for example, there's this bacteria called Akkermansia municipala in the gut, and they're known to maintain the mucus layer. So that's been researched as a way of improving mucus layer health in different disease settings like obesity, diabetes. Non-live biotherapeutics is using dead bacteria or dead microbes or even their metabolites as a way of changing the gut microbiome. I'm going to dive in a little bit deeper to phage therapy and synthetic biology. So phage, they're viruses and they're naturally in our gut, they're everywhere. How they multiply is through injecting their genetic material into a bacterial cell. So when this happens, there's two ways of them multiplying. The first way is that they hijack the bacterial cell, multiply inside the bacterial cell, and when they release, they kill the bacterial cell. So that can be potentially used as a way of eliminating unwanted bacteria from the gut. The second way these viruses multiply is through injecting the genetic material to bacterial cells. The bacteria incorporates that material into their genome, and the bacteria multiply. So there's new genetic material that's been introduced to the bacteria. And this can potentially be used as a way of enhancing the fitness of the gut microbiome, introducing things that we want, more to happen in the gut microbiome. Therefore, introducing this, rebalancing the microbiome through eliminating what we don't want and enhancing the beneficial effects. Synthetic biology is where we are editing the genetic material of microbes. There's multiple applications of this that's been studied in terms of gut microbiome research, but the three things that I would like to touch on is we can edit the genetic material in probiotics for them to function better, for them to, for example, potentially in the future to sort of tolerate stomach digestion or small intestine digestion, for them to go to the colon and adhere better, stick to the colon better. Like I mentioned with phage therapy, there's potential of synthetically engineering phage viruses to introduce a targeted change to the microbiome. So we can perhaps engineer different phage viruses carrying particular genes that will then introduce those genes into our gut microbiome, the bacteria, that will then carry these traits. Say, for example, if you want the microbiome to produce more butyrate, maybe this is a way. Going back to the concept of microbial consortia, so it's still, ideas, and there's research being done on this, but building microbial communities, synthetic microbial communities that do a task that we want the community to do together is another application of synthetic biology in gut microbiome research. So like I mentioned earlier, these last two approaches, phage therapy and synthetic biology, is not really being is not available as a therapeutic yet, but there's a lot of research needed for us to get there. But these are where the field is headed to, in addition to using prebiotics, probiotics as personalized ways to change the gut microbiome. Okay, so with that, I'd like to thank FAAB. I'd like to thank my mentors and all of you for coming along, and I'll be more than happy to answer if you have any questions. Thank you so much.
I'm just wondering, have you and your team considered vegans, vegetarians? Do they get all the microbes they need for their gut metabolism with the foods they eat, or they've missed some to make certain vitamins and things? Have you done any work with that yet? Or?
Yeah, so there's a lot of studies looking at that actually. So depending on the diet, obviously the gut microbiome do vary. Vegans and vegetarians, they do have a lot of dietary fiber digesting bacteria, where largely animal-based people who are having an animal-based diet, they do have fat metabolizing bacteria, etc. So I think either end. So vegans do have a lot of important gut bacteria that can produce short-chain fatty acids, and in terms of gut health, have, I guess, better fitness because there's more metabolites that's being produced. So I think as a rule of thumb, it's best to have a balanced diet, have more vegetables, have more dietary fiber, and like when the diet is really high in animal food products, it's not really good for the gut microbiome or the health.
But it's not essential to have animal products then?
Sorry?
Is it not, it's not essential to have animal products then, you feel, or what research
Well,
seems to indicate?
For gut microbiome, not so much, but overall health, yes, like, milk and all that, and even protein is quite important. So I think my personal view here is, having a balanced diet is the key.
Yep, of course.
More lean meat and having a
I agree.
balanced diet.
Yep. Thank you, thank you.
Over here.
Hi, two questions. First question, a while back, I can't remember who mentioned it, but you mentioned something to do with lactive something, can't remember. But does that have something, does that have something to do with lactose intolerance? And if so, how does that affect the microbiome? And like, why does that happen? And I can't remember my second question, never mind.
Good. Is it lactobacillus? Right. Not really. So lactobacillus is really important in the early life to digest milk products. And it is, like we discussed before, it's been used in probiotic products. But partly because it is one of, Lactobacillus bifidobacterium being the very first bacteria that's been studied due to lack of all the techniques we had. But yeah, no, there's definitely microbiome differences when it comes to lactose intolerance, but there's no real correlation, although the name sounds pretty similar. Yeah.
Is there a point where the gut
Sorry.
can't...
Yes, please. Yeah.
Is there a point where the gut can't be fixed? And if so, what diseases would that evoke?
Yeah, that's a really good question. So yes, there could be a point that the gut can't be fixed. So, if you're sort of having really poor diets, that's low in dietary fiber, that's, high in processed food. And if it happens continuously, using techniques like prebiotics and probiotics, it becomes difficult to fix it. Because if we don't have the bacteria that's needed to digest, say for example, dietary fiber, no matter how much dietary fiber we intake, it's going to be difficult to fix the microbiome because we don't have the microbiome to consume them. So yeah, at some point, as the microbes disappear from the gut, it will become pretty difficult to sort of reverse it. It's also known that's safe for things like antibiotic usage, right? So gut microbiome can change during the consumption, but it also has that ability to restore itself if the microbes are present within the community. But it really don't go back to its original composition. So yeah, thanks.
Question for Dr. or soon to be Dr. Pereira. Given the degradation of the probiotics that you put into your system as they pass through the stomach and the small intestine, how much of this stuff do you have to take to get a material increase in the microbiome, in the product in the microbiome?
Yeah, okay. So usually the products that are available in the market, they have loads of bacteria in there. It's like billions of them. So I have tested some of them in my previous work. So it's usually 10 to the power 10, let's say, bacteria in the product. And as per TGA, at least 10 to the power 6 should get there, which is the case. But then because the conditions are so harsh, they might not be able to fight the pathogenic bacteria in the gut and adhere to the gut epithelial. So that's where we are trying to add some more stuffs like the prebiotics so that there is some sort of feed. So once they get in there, it's like a protective covering. So hopefully most of them get over there. Yeah. Hopefully that answers your question.
Yeah, okay. My question is with regards to fecal transplant. With the screening process that you talked about with fecal transplant, what is the current thinking with the screening on mental health? So we know that there's the connection. So particularly around mental health, Parkinson's disease, what have you. Has there been more work done on that and the thought of fecal transplant carrying these diseases into another person?
Yeah, that's a real concern. At the moment, most of the screening is based on the host. They screen the host, not so much the microbiome. Now that we know, I think it is a really good point that has to be incorporated in the screening that's been done, because we do know that transplanting just the microbiome can really induce the disease. For example, like obesity or even depression and mental health, just transplanting the microbiome of people with the particular disease to, say, a mouse model, for example, just that can cause the disease in the mouse model. So the microbiome can actually carry that information. So yeah, I think it's a really good point that's to be tested.
Firstly, thank you so much to all the presenters. It's been a fantastic range of presentations tonight. It's been really very interesting. My question is is there a lot of research around the diets of blue zone occupants, have really healthy lifestyles and live longer?
I couldn't hear you just then.
Sorry, is there a lot of research in the diets of blue zone occupants, those communities where people live very long lives?
Yeah.
The sort of diets that they have, are they particularly good for are they particularly representative of having good microbiomes in those communities?
Yeah, I mean, there's research on that quite a lot, especially these Japanese communities, who sort of live beyond 100 years, etc. There is a difference in their diet. They actually obviously eat more, dietary fiber, more veggies and fruits, and fermented food is another thing. So there's also another key thing is there's studies showing that the timing of the day that they eat also has an impact. So they've shown that, people who eat, during the the light cycle and fasting to have an impact on, healthy life. Yeah.
Hi, thank you all. Just two quick questions. Very early on, you talked about the percentage of bacteria in a human, that 43. I've been reading studies as recently as 2021 suggesting that it's upwards of 90. So the question is how do we measure it and why the disparity?
Yeah.
And number two, with regards to the connection, usually neurological, from the vagus nerve, from the brain stem into the gut being considered more of a sub-brain to our primary two brains here. The testing that you do between transit time as a predictor for Parkinson's, what screening processes is that? What are the screening processes in practical land? So I go to the doc, my doc goes where to get me assessed, and where is that down the pathway of Parkinson's as a disease progress?
No, thanks for those questions. Yes, to answer your first question, things keep changing. The very first sort of reference to, there's 100 times more microbes compared to our human, cell counts. It was really based on a really old study, so it wasn't really accurate. And this sort of reference of around 43%, it's around the ratio is pretty similar. It's sort of updated recently, around 2016 or 17 to that. So it really comes down to the availability of techniques that sort of count. And it also varies from individual to individual. So it's really a range that we're talking about. It's really not, an exact number. And this is sort of in average in a healthy adult that's around 70 kilo. It's really a range, and it really varies significantly between people. To answer your second question, so the gut transit time can be measured using dyes. So this was done in mice, but it's the same in people. So there's dyes that we can't metabolize, so they're not being absorbed in the colon or the small intestine. They just sort of traverse with food. So we take that dye and sort of wait for the color change to appear in feces. And in terms of the timing, with Parkinson's disease, it's been shown like 10, 15 years, so it's pretty early on. With the particular disease that we are looking at, we really haven't taken into humans, because there's a difference, because what we see here is the transit time is quicker. Whereas in Parkinson's disease, the transit time is slower. So there's more constipation that these patients experience really early on. We really don't know why the difference is, and we are looking into this more. But yeah, so I think years in advance, if there's a risk factor involved, yeah.
Hello, two questions. So the first one is about children born via cesarean section versus natural delivery.
Yeah.
So because the ones who are born by cesarean do not pass through the birth canal, and so are there ways of, like, apart from fecal transplantation, are there ways to kind of give the baby the mother's microbiome?
Yeah.
Breast milk or anything, or So second question, there are some products, probiotic products, that claim that they have some shields around the bacteria within the capsule that can pass through stomach acid. So I don't know how true those claims are. Is it just a money-making thing? Or is that like, there's existing technology already in place that we can get maximum benefit from these probiotic products? Thank you.
No, thank you. So to answer your first question, there is vaginal swabbing that happens with cesarean section born babies as a way of introducing the microbiome to them. Also, when it comes, so it's mainly lactobacillus that they get exposed to, different strains of lactobacillus. And they are available in, not the exact ones, but lactobacillus as a generic, like a group of bacteria. They're available in breast milk too. And also in current formula, they've been added. So there are ways around it. And there's studies showing that this microbiome difference really sort of solves itself as the baby's age. So it's not, there are impacts of that, and they've shown, health outcomes related to that, but largely, yeah, so, right, yeah. So there's studies showing that in terms of immunity, there's lower immunity. There's, in terms of allergies, there's high prevalence. But having said that, there's also studies showing, it sort of solves on its own, within 5 to 10 years of life. To answer your second question, yes, there are technology that can sort of shield the individual bacteria. How efficient they are, I'm not really sure, to be honest, and we haven't really looked at it. But these technologies are being developed as sort of ways in comparison to without those shields for maximum numbers of bacteria or these probiotic bacteria to reach the colon. So I don't think they're all just money-making strategies, but you have to sort of take it with a grain of salt. Like because when it comes to marketing, I think it's really important to sort of also look, the claims are backed up with research, and that's what we're trying to do, especially working, with already available products to see whether, these claims are true.
I think you've answered enough, but maybe the last one, since I mentioned it.
Sorry, just a really silly question. You showed a picture of how you were doing the culturing outside of the body, but how do you actually get the sample of the microbiome?
Yeah, no, that's a really good question. So the first thing is feces. So feces actually carry the gut microbiome, the ones that are sort of like being flushed out of the system. And there's studies comparing the fecal microbiome with what actually is in the gut. And they're not exactly similar, but in terms of composition, the story sort of, like looking at the impact of a disease or a prebiotic, probiotic, it tells more or less a similar story. The other way is through biopsies. So with colorectal cancer, for example, we do work with human biopsy samples. So those two things being the main ways. But when it comes to people, usually the first thing is fecal samples.
Well, it's fascinating. Some of us have the pleasure of being able to ask Hasinika and her team questions all day long at work. So we could be talking for a long time, but I think they've had enough. So please join me in thanking Joanna, Lauren, Anna, and Hasinika. And thank you for your attendance.